And our final talk of the session before lunch will be our first talk by the home team from the Allen Institute. And that will be Amanda Haupt, my esteemed colleague. Amanda's talk will be titled Endogenous Gene Tagging Illuminates Cell Organization. Thank you.
Thanks, Brock. Thanks again, everyone, for being here. We're just really proud to host this event every year for year number 2. This time last year, we had just released our first 5 cell lines out to the public. Since then, so by the end of this month, we'll have 20 released, and then a plan to have another 15 next year. So I'm excited to give you an update on all of that we've been doing. So you've heard this a couple times now already, but some of the things that we rely on here at the Institute primarily rely on these human-induced pluripotent stem cells. So this is where we have a donor who provides a cell. We can then reprogram them into pluripotent state, which can then be differentiated into many, many different cell types that have a wide variety of applications, but also that we're using here to study basic biology. Now, we don't do the reprogramming state, but we do this differentiation state. Rick mentioned this morning that the first cell type we're looking at is the cardiomyocyte. I'm going to tell you a little bit more about that in a few slides. The benefits of these, they're human, they're diploid, they're proliferative, and they have this differentiation potential, which allows us to edit the stem cells and then look at many different cell types from that edited line. And again, this is going towards our goal of really understanding the normal human cell. The other technology that we rely on is CRISPR-Cas9, which is, of course, the molecular scissors, which allows us to very specifically insert a piece of DNA, in our case, a piece of DNA that encodes the fluorescent protein, into the host genome, either right at the beginning or right at the end of a protein that's already being expressed in the cells. And this gives us an endogenously regulated fusion protein, which is then ideal for live imaging. Rick gave you a little bit of an overview this morning, but just to put my team into context, we are really the start of this pipeline. So it's really important that we're creating these very high-quality cell lines that are going to be used by our microscopy teams to develop the images that are going to go into the models and the animated cell tools that we're also developing. How we do that, we're of course using CRISPR-Cas9. We use the RNP transfection method, which we believe is a little bit more gentle, allows us to generate these cell lines. When we transfect, we end up with an edited pool that's maybe 1% GFP positive. It's not highly robust, but it's enough that we can enrich for these cells by FACS. And then we go on to generate clonal cell lines. What we're really looking for is either a monoallelic or biallelically tagged cell line that we can then run through our whole gamut of quality control, Western blots, morphology, all of these things, so that we end up with a cell that we can then put out for distribution, that we can do our live cell imaging on. And then sort of something that we've been really tackling this year is this differentiation aspect, where we're turning the cells into cardiomyocytes and then also feeding them back into our live cell imaging platform. This year we published a paper which is available on our website. I just want to highlight a couple of the things that we learned. And basically, the genome, especially in stem cells, is editable. To date, we've done 30 unique loci with pretty good success. We are very careful about our CRISPR design that we don't choose promiscuous CRISPRs. But one of the things that we've reported was that we have not detected any off-target mutations. But what we do find is actually random integration of our donor plasmid. We can detect this through a ddPCR assay. And then we also find on-target misrepair. So when the cell is incorporating that GFP tag, sometimes it does it wrong. So it's really important to look for these things if this is something that you're interested in doing. We identify these with both junctional PCR and Sanger sequencing. And then also, because we're sort of looking for a lot of monoallelic genotypes, we also check that wild type allele and make sure that the Cas9 hasn't introduced any mutations on that allele also. So I just want to give you a quick tour of the website. If you went to allencell.org and then went over to cells and the cell catalog, you can see a list of all of the cell lines that we have available for distribution. This gets updated regularly. If we click on one of them, I'll choose the nuclear lamin B1. So this is going to be the nuclear envelope. The first thing that you're going to notice are these really beautiful Z-stacks that our assay development team has provided to you. So going from the bottom of the cell up through the top, you can see that beautiful nuclear envelope. This is all live cell imaging with these endogenous tags. So there's no artifacts, nothing getting in the way. You can also look at these really amazing time-lapse movies of the colonies. So these cells grow in these nice colonies. You can see them dividing and sort of moving around a little bit. So these are just a couple examples of the images that we're getting from these cell lines that we've generated. if we go to the bottom of the page, my timing's not great here, sorry. You can see the editing design, including the CRISPR RNA sequence that we used to generate the line and where we've inserted it into the genome. We go over some of the genomic characterization to make sure that that GFP is incorporated on target, and then some of the protein analysis that we do also, some IF and Western blots. We also want to make sure that our stem cells are maintaining their pluripotent state, so we do a bunch of quality control in that regard as well. Additionally, on our website, you can find protocols. If you're interested in obtaining the cell lines, we will help you grow them by providing a protocol. Also, if you wanted to do this type of transfection, we have a protocol out for that also. You can obtain the cells through our distributor, Coriell, and they also give you information about the protocol on their website. Additionally, if you're interested, again, in doing this in a different cell type, we'll distribute our plasmids through Addgene so that you can have the donors. And then finally, if you're interested in learning more about the way that we're doing our quality control or generating the cell lines, on our website under research and publications, you can find the paper here. Okay, so once we have these cell lines fully validated, then we can really use them, and it's been really amazing. So here's an example of our alpha tubulin tagged line, which is showing the cytoskeleton with this beautiful mitotic cell here. And like as I mentioned earlier, one of the things we've been really working on this year is this differentiation into cardiomyocytes and seeing how these structures change from the stem cell into the cardiomyocyte cell. So here's the cardiomyocyte cell. So we took this cell line, differentiated it, and now you can see these microtubules have really reorganized in these cells. And of course, again, this is live cell imaging, so we can actually watch the cells beating in basically real time. So that was an example where the stem cell was expressing the gene that we had tagged and also was expressed in the cardiomyocyte. But there's many genes that we want to study that are not expressed in stem cells. So here's an example of a cardiomyocyte-specific gene that we have been able to tag. So now this is a little higher magnification. So this protein is actually in the sarcomere. And you can see those nice sort of railroad track-like units as the cardiomyocyte is contracting. So we're really excited to see what kind of data we can get out of these images, and more to come on that for sure. Another way that our stem cells are being used in the community, this is a figure from the Friedman lab. These are kidney organoids, sort of at varying degrees of magnification. This is that lamin B1 nuclear envelope cell line. And here's our cell-to-cell junctions cell line. And then this is, of course, the cytoskeleton with a nice example of a dividing cell. So this lab's really interested in studying kidney disease, and the use of our cell lines has really enabled them to understand what's going on in the cell-to-tissue level, and not only understand how the tissues are functioning, but hopefully also lend information about how the diseases are happening in the tissues also. So finally, just want to say one quick thing about something that we're starting to work on. Whenever we say that we're editing stem cells, people always say, okay, well, you've got these 1 edits, can you put more edits in? And the answer is yes. We have an example here of the tubulin line again with this beautiful dividing cell. And in the same cell line, we've also tagged the endoplasmic reticulum. So here's that channel. And when we merge them, we can see now 2 endogenously regulated proteins in real time in live cells at the same exact moment. So of course, having these multiple edit lines is going to give us more powerful understanding of the biology, and also hopefully feed into our modeling pipeline and give them some more data to work with. Later today, you're going to hear Dave talk about how we're taking the individual cell lines and actually integrating them into an integrated cell. So hopefully you guys will be around to catch that later today too. So that's it. I'll let you guys get off to lunch. Just want to thank my team and thank everyone again for joining us today here at the Institute.